Hi, everyone. Thank you for attending E-IPER's very first ever virtual defense. This is not how I imagined wrapping up my time here at Stanford. I'm sad not to be presenting in person in Y2E2 299 today, but the silver lining is that I have a lot more friends and family able to attend virtually today than would have been otherwise possible. So I'm thankful for that and glad that you're all here. Today I'll share with you some research that I've been doing over the last 5 years on the human environment dynamics in the eco-epidemiology of schistosomiasis. But first, what possessed me to do this PhD in the first place? Before coming to Stanford, as Giulio mentioned, I spent 4 years chasing mosquitoes in urban Atlanta, first as a master's student in epidemiology, and then as full-time research staff. Mosquito-borne diseases were fascinating to me because their transmission is affected by the environment. That is, temperature, rainfall, and water chemistry all affect how mosquito populations grow and how they transmit disease. I was enthusiastic enough about my work that I climbed into manholes to deploy temperature loggers, like you see here, to measure the conditions where mosquitoes were breeding. I was fascinated by how environmental processes could influence risk for human disease, but I also began to think about how people were part of these systems too, and I wanted to understand what role they played. And so this became the focus of my search for a PhD program and how I ended up at Stanford. So I'm going to start out with this aerial image of a water access site in a village along the Senegal River. This is one of the 16 villages where I've worked since starting to work with Giulio and Sana in 2015. In this photo, you see a wide variety of activities occurring in and near the water. There's a group of boys swimming, playing, and splashing. There are people wading into the water to collect water in buckets. There's a man washing a horse. And there are people on the edge of the water likely washing dishes. On the shore, we see a couple of fishing boats, as well as a group of women sitting on the shore washing clothes with water they've collected from the river. These are all typical activities for a village like this, but there are also activities that expose people to the parasites that cause schistosomiasis. And schistosomiasis is a disease caused by parasitic worms that come from the genus Schistosoma, and so we can call them schistosomes, and they look like this under a microscope. And depending on the parasite species, these worms inhabit either the urogenital tract or the intestinal tract. And if left untreated, these infections can last for several years, on average 3 to 5 years. But with prolonged or repeated infection, severe and chronic disease can develop, including kidney and liver damage, cancer, and even infertility in women. And for a disease that most people haven't heard of, schistosomiasis takes a huge toll on people living in tropical latitudes. It is second only to malaria in terms of the global burden of parasitic disease. There are nearly 800 million people at risk worldwide, and an estimated 200 million are actively infected. Those people live mostly in sub-Saharan Africa, but Latin America and Asia are also, parts of Latin America and Asia are also infected. And in 2016 alone, schistosomiasis caused the loss of 1.5 million disability-adjusted life years on affected populations. And so there's 5 species of human schistosomes, 5 different parasite species. But today we'll focus on the 2 that affect our villages in Senegal, and that is Schistosoma mansoni, which causes intestinal schistosomiasis, and Schistosoma hematobium, which causes urogenital schistosomiasis. And I mentioned that people doing their laundry and washing their animals in the water was exposing them to schistosomiasis, schistosome parasites. And that's because these parasites have a complex transmission cycle. They're transmitted by freshwater snails, and the parasites will exit the snails as free-swimming larvae that can penetrate the skin of people who are in contact with water. And so this is an unusual route of transmission for a human parasite. It's not like mosquito-borne transmission where a mosquito is actively biting a person in order to transmit the parasite. The snails are passively shedding parasites into the water. And schistosomiasis is also not like most waterborne pathogens. You don't need to drink the water to become infected, just be in contact with it. And so infected people contribute to the completion of the life cycle by excreting parasite eggs in their urine and their feces. And the eggs that reach the water, again, will hatch into a new larval stage that can infect the snails and begin the cycle all over again. And despite the complexity of the transmission cycle, we combat schisto with a relatively simple intervention, and that is deworming or mass drug administration programs. And these often take place in schools where all of the children in a school are presumptively treated on some regular basis. And this can be yearly, maybe twice a year, every other year, depending on how high the prevalence is in a place. And these programs can prevent the development of chronic and severe symptoms, and they have decreased the burden of disease in many of the places where they've been implemented. And because of the impact that they have on reducing the infections in infected people, mass drug administration campaigns are considered highly cost effective. They generate a lot of health benefit for the money invested. But mass drug administration campaigns have one key shortcoming, and that's they don't prevent reinfection after treatment. And there's two main reasons for this that are apparent in the life cycle itself. First, snail populations will remain in the water or in the environment despite treatment, and people will likely continue to come into contact with water. And as long as both of these processes continue, reinfection will be common. And so these processes of human interaction with an environment that supports parasite transmission frame my dissertation research, whose overarching question is, how do social factors influence the ecology of schistosomiasis? And I address this overarching question with three specific chapters. And the first explores local perception of schistosomiasis risk, the lived experience, asking the question, how do people perceive the places where they come into contact with water? And how do they perceive their risk for infection in these places? The second quantifies the relationship between agricultural livelihoods and schistosome infection, asking the question, does the presence and intensity of infection in school-aged children increase if their household cultivates a larger area of irrigated land? And the third examines the relative contributions of three distinct but interrelated components of risk, and that is exposure, hazard, and vulnerability. And what are the relative contributions of these three things to schistosome infections? But first, I'll talk a little bit about the geographic context where I've done this research. And first of all, schistosomiasis is particularly problematic in settings where the environment has been modified by water resources infrastructure, such as dams or irrigation schemes. And of the 800 million people that are at risk for schistosomiasis, 100 million of those live in close proximity to dams or irrigation infrastructure. And meta-analysis across many different settings shows that risk for schistosomiasis doubles when you live near a dam or a reservoir. It similarly increases near irrigation schemes. And that's because dams stabilize water levels and create new and better habitat for snails. And you can see from this photo, what was once a meandering river now has a massive lake in the middle of it. And that's a lot of real estate for snails to occupy. But these dams also create opportunity for agriculture and increase human interaction with the environment. This image is actually a satellite image of the Monintali Reservoir in the upper basin of the Senegal River. It's located in Western Mali here. And this dam produces hydroelectricity for the capital cities of Mali, Mauritania, and Senegal. It's conceived in tandem with the Diama Dam, which is located in the lower basin of the river, just 30 kilometers inland from where the river empties into the Atlantic Ocean. And together, these dams were constructed to reduce vulnerability to climate change and drought. And while the Monintali Dam is a hydroelectric dam, the Diama Dam is a saltwater barrier, and this prevents saltwater intrusion upstream and supports agriculture in the lower basin. And so the lower basin is near Diama Dam is also where our field sites are located. So I'll be focusing on this region of the basin for the rest of today. And here you see a satellite image of the lower basin before and after construction of the Diama Dam in 1986. And before dam construction, the river flowed through an otherwise pretty arid landscape. But since dam construction, that landscape has become a lot greener. In fact, you can actually see exactly where the dam is because everything upstream of it is bright green or dark green, whatever you prefer. And so this reflects both growth of both aquatic and agricultural vegetation and illustrates how much the dam transformed this landscape. We also see enormous increases in cases of both types of schisto after dam construction. And these levels of infection still remain shockingly high today. And that's 30 years later. So this dam was built before I was born, and the infection is still very high. So these processes of land use change and disease occurrence have happened over the same space in the same time. And the satellite imagery makes the environmental change obvious, but I wanted to understand the human side of this story. How are people interacting with their transformed environment, given the economic opportunities that the dam created? And we investigated these questions in 16 rural villages in this region. I mentioned that the schisto prevalence was high in this area today. At baseline in 2016, 77% of the school-aged children were infected with the urogenital parasite S. hematobium, while 32% were infected with the intestinal parasite S. mansoni. This is also an agricultural area where we have most of the households engaged in some sort of agricultural activity. But as I get into the first chapter of the dissertation, I'll focus on qualitative data we collected in just 4 of these 16 villages. And in these 4 villages, we wanted to understand how people perceived their interaction with the environment and their risk for schistosomiasis. So we gathered people to talk about their perceptions in focus group discussions. And in each village, we conducted 3 separate discussions with gender and age stratified groups. And these groups ranged in size from 5 to 9 participants, and we had 74 people participate across all 12 groups. And each discussion lasted about 1 hour and 20 minutes, which yielded 16 hours of audio in the local language, which eventually got transcribed to 300 pages of text in French. And so a research assistant and I coded these transcripts together in a software called Deduce, and we used a codebook that we developed deductively, but revised inductively as we came across new topics in the data. Then I took that coded data and used thematic analysis to identify key findings from these conversations. And this analysis revealed 3 main findings. First, people are knowledgeable about risk for schistosomiasis in their environment, and they use their knowledge to reduce their exposure, adapt their behavior, but their local circumstances ultimately keep people in contact with water. And I'll go into detail with each of these findings in the next few slides. First, we found that focus group participants across all 4 villages had a sound understanding of risk for schistosomiasis in the environment. They commonly linked their risk for infection to their contact with river or lake water. And this 53-year-old man said, "It is there at the river that schistosomiasis enters your body. If you're told something else, know that you are cheated. It is there that you feel it enter squarely into your body." And so people recognize not only that parasites were present in the environment, but that infection results from the water contact activities that they undertake on a regular basis. And while no one in any of the groups mentioned specific biological details of the transmission cycle, it was common for people to associate their risk for schistosomiasis with high temperatures at midday. And this 15-year-old boy said it this way. He said, during the heat, we can get infected with schistosomiasis if we go to these water points and around noon and beyond during the heat when the sun is at its zenith. This actually reflects known dynamics of schistosomiasis in the snail host. Snails tend to shed parasites into the water around midday. And so this perception, though not grounded in specific knowledge of the transmission cycle, is grounded in an epidemiologic reality. And focus group participants also described their efforts to adapt their behavior to the risk that they perceived. A young woman from one of the lake villages reported, "Now at 1 p.m., if you go to swim there, the parasite can get into your body. That's why sometimes we have the habit of bathing in the morning or in the evening, around the 5 p.m. prayer time or later." And so this woman is also making reference to the time of day, and she perceives the risk to be higher at midday, and she's talking about adjusting her behavior and going to take a bath in the lake at a different time of day when she perceives the risk to be lower. And so these reported behaviors reflect earnest efforts by people to mitigate their risk for infection. But almost always the focus group discussions returned to a sense of inevitability and unavoidability. People have very few alternatives. The difficulty of avoiding the water was expressed by this 68-year-old woman. She said, "Water contact's not good for you. It's not good, but we have no choice because we are near the lake. If you say, I will not do it, it is certainly not possible. It cannot happen like this. Fortunately, we pray for ourselves and we are given medicines, but we cannot protect ourselves. We are not easy to protect." So avoiding contact with water is not feasible in this woman's village, even though she knows it can cause harm. And she talks about the medicines that get distributed. And this is what the Mass Drug Administration campaigns that I mentioned earlier. These help, but she doesn't view them as a solution. They're not preventing new infections from happening. And so the perceptions that focus group participants shared indicate that people are aware of the risk in the environment, but they face an unfortunate trade-off, having to choose between their immediate need for subsistence and carrying out their daily lives and the long-term health consequences of infection. These findings make clear the importance of finding ways to reduce the risk of acquiring infection from the environment when exposure is unavoidable. But people are knowledgeable, and that knowledge translates to action. And the initiative that people reported taking to reduce their exposure gives me hope that coupling community engagement with environmental management can reduce the burden of disease in combination with Mass Drug Administration. And so before I move on to the next chapter, I want to pause to acknowledge that the dissertation research I'm presenting today is nested in a much larger project. And the next chapters will involve the integration of multiple data sets collected by multiple teams. So I want to quickly review those now. First, the medical team at our partner organization, Espoir pour la Santé, collected 3 years of parasitology data from school-aged children in all 16 villages. This involved collecting and processing urine and stool samples for over 1,400 kids to determine if they were infected, as well as how infected they were. How many eggs are we finding in their stool and urine? This team also treated kids after each round of data collection, both to cure them of their infection, sort of a Mass Drug Administration round, but also to help us measure reinfection after treatment in subsequent years. Then an ecology team led by colleagues in the DeLeo lab conducted exhaustive snail sampling of the 32 water access sites in all of the 16 villages. This team quantified snail habitat using drones. They collected a ton of snails while wearing waders in 100-degree heat, and then brought snails back to the lab to identify them and determine whether they were infected. Finally, I led a team of colleagues at EPLS in collecting data on more of the human dimensions. And this involved collecting survey data from all of the households where kids in the parasitology study lived, as well as the focus group discussions that we discussed in chapter 1. I designed the data collection instruments and joined the team in the field overseeing data collection and data quality checks. We collected all of these data with the intention of being able to merge and integrate them, the different data sets in our analysis. So in chapter 2, I combine 2017 parasitology data with household survey data from 2016. And so in this chapter, we shift from the lived experience and local perceptions of the environment to quantifying risk as it relates to agricultural livelihoods. And here we are again at this water access site in this village. And sites like these are often the focus of research on schistosomiasis risk. And that's because these are where most exposure happens. But they're not the only places where people are coming into contact with water. And I'm going to use a few more aerial photos to illustrate how large the relevant scale of exposure might be. In this photo, you see a village water point outlined here in beige with agricultural fields along the shore. There are also a couple of irrigation canals here and here. And it's clear that these canals serve the fields that you see, but they also extend beyond the frame of the photo. And so water is getting transported to fields that are farther inland than we can see. And that means that exposure could be happening far beyond the water access point that's in that beige box. And this is another example in a village where we did focus groups. So we learned some additional nuance about how people interact with their environment in this particular village. So the village is down here, so are its water points. This is probably where most exposure is happening. But there are a set of garden plots nearby, which are directly adjacent to the stand of cattail reeds along the river. And in these plots, people will come into contact with water when they cut back enough reeds to access water and use it to irrigate the crops. Now, they'll either do this with buckets or maybe with diesel pumps. But what's striking about this image is that rice fields take up an entire third of the frame. These fields are a direct result of dam construction and are served by irrigation infrastructure that's drawn from the river. And we visited these fields following a focus group. They're a couple of kilometers away, and so it's a very long walk and not quite as long drive. But we learned that people sometimes spend the night in these fields to protect their crops from birds. And when they do this, the canals are their main source of water to meet their various needs while they're out there. And so between the rice fields, the garden plots, and the village itself, exposure is likely occurring across an enormous area. And so places like this that look like this are likely a non-trivial source of schisto exposure. We know that schisto is often associated with agricultural landscapes, but most studies that have demonstrated this relationship have done so with village-level aggregations of disease and agricultural activity. These studies support the idea that increased risk for schistosomiasis has to do with environmental transformation. But we were interested in drilling down to the household and individual levels to understand whether the household engagement in irrigated agriculture is associated with infection. In other words, are the livelihoods that people are undertaking that were made possible by the dam compounding the risk that dam construction has already created? So in this study, we investigated whether schistosome infections increase in school-aged children who live in households that cultivate larger areas of irrigated land. And to answer this question, we used data from over 1,200 kids in the parasitology study. We combined this with survey data from all of the households where those kids lived. And with that survey data, we calculate the area of irrigated land reported for each household. So irrigated land is our main explanatory variable of interest, while infection is the outcome. And because we're looking at both infection presence, so whether or not a kid is infected, as well as infection intensity, or how many eggs we're finding in the sample that they gave us, we use two different types of statistical models to quantify the relationship between infection and agriculture. And in looking at this relationship, we also control for demographic covariates that were derived from the survey data, as well as nested structure of the data itself. And here I'll show estimates that we generated from models of infection presence. And I'll have urogenital infection on top and intestinal infection on the bottom. The x-axis represents values for the odds ratios that quantify the relationship between irrigated agriculture and infection. And the vertical line that you see represents an odds ratio of 1, or the value at which there is no association between agriculture and infection. I'll populate this space with point estimates and confidence intervals, and we'll look at their relationship to this vertical line. So point estimates and confidence intervals that occur to the right of the line, we can interpret as increased infection with irrigated area. And if it's to the left, it'll mean that the infection decreased with irrigated area. But if a confidence interval crosses the vertical line, we won't be able to draw a conclusion either way. So I'll go ahead and reveal the estimates for urogenital infection presence. And you see that all three are completely to the right of the vertical line. And this reflects an increase in infection. I'll highlight the best fitting model here and show you the numbers. And the odds ratio of 1.14 represents a 14% increase in the odds of infection with every hectare of land a household reports. On the bottom, we see the estimate for intestinal schistosomiasis, is very close to 1, and its confidence interval definitely crosses that vertical line. So we conclude that there is no relationship between irrigated land and intestinal schistosome infection. Now we shift to models of infection intensity, or how many eggs we're finding in children's urine and fecal samples. And these estimates are a result of a different statistical model, but the interpretation of the plot is similar to the last slide. And so we'll look at point estimates and confidence intervals in relation to the vertical line. Across the board, we see confidence intervals do cross that null value of 1. I'll highlight the best fitting models here with the numbers, but we conclude that there is no association between infection intensity for either infection. And so from this, we conclude that the presence of urogenital but not intestinal infection increases with irrigated area reported by a household. And this is a relationship that's been demonstrated before, but we do so at a finer scale than has been done previously. And with this finer scale analysis, we support the notion that social factors and finer scale kind of behavioral and economic processes, namely that agricultural livelihoods, increase risk for urogenital schistosome infection. It's not just the environmental change created by the dam. And this also points to the potential importance of agricultural water sources and the role they may play in sustaining transmission. This means that focusing only on the village water access sites may be missing important pieces of the puzzle and may be missing important opportunities for intervention. And so in chapter 1, we learned about how people perceive there to be an unavoidable risk of schistosome infection in the environment. They have both knowledge and agency to adjust their behavior to the risk they perceive, but they live in a place where daily life is tied to the environment, and that makes it really difficult to avoid exposure completely. And in chapter 2, I show quantitatively that the predominant livelihood in this setting and the one that the dam was designed to support increases the likelihood of infection with urogenital schistosomes. Now we return back to this water access site one last time in order to think about schistosomiasis risk as a combination of 3 different but interrelated concepts. So we know water contact activities like we see in this photo are the main route of exposure for infection. It's how people come into contact with parasites. But that water contact isn't inherently harmful. I grew up in Minnesota swimming in lakes without having to worry about getting infected with schistosomes. It's the water contact in the presence of a hazard, or these snails that are maybe hanging out in this floating vegetation here. That's what makes water contact harmful. Similarly, water hazard doesn't matter unless there are processes ensuring some baseline level of exposure, and that's where vulnerability comes in. Whether people have alternative ways to meet their daily needs determines the consistency with which water contact occurs. And as these conditions converge to determine risk, I wanted to understand the relative importance of each component to both infection presence and infection intensity outcomes. And to answer this question, I draw in 3 data sets from the larger project. So this involved parasitology data from 2018, snail ecology data from 2017, and household survey data from 2018. And I build on analysis that was recently published by the snail ecology team. They found that the best environmental predictor of infection in the kids in the parasitology study was area covered by snail habitat, or the non-emergent vegetation that's outlined in this aerial photo. And so I build on these findings by incorporating indices of exposure and vulnerability calculated from the survey. I'll quickly summarize the different indices that we calculated here. And similar to chapter 2, we use individual level infection data from our parasitology study as our outcome, looking at both presence and intensity of infection. And I use both survey and interview data to measure exposure in terms of water contact. And this involved the development of 4 different metrics, ranging from a simple recall of visits to the water access site. So how many times in the last 7 days did this child go to the water access site, to more complex metrics that deal with specific activities and thinking about the frequency, duration, and extent of exposure that happens when doing those activities, summed across all of the activities we considered. And then we measured hazard using the ecological data, as I mentioned, based on 6 different metrics. So all of these metrics are based on area of snail habitat as the best environmental predictor of infection from that previous study, but we formulate this in a few different ways. Some of these metrics involved only the site that was nearest to a child's household. Others involved the habitat area across the entire village. And similarly, we looked at habitat area just at the peak transmission season, as well as over the entire year. And then for those indices where we used just the nearest site, we adjusted those for distance between a child's household and that water access site. Lastly, vulnerability metrics centered on the kinds of water and sanitation infrastructure reported at the household level. And this includes how much a household relies on surface water for different activities, whether members of a household use private versus shared toilet, or maybe had no toilet at all, and an index of socioeconomic status that was based on assets reported by each household. So we set up statistical models that were similar in structure to the ones we used in Chapter 2. We used the same kinds of infection outcomes, as well as a similar set of covariates and nested structures. But our main explanatory variables of interest are the exposure, hazard, and vulnerability indices that I just walked through. We started by fitting models with a single exposure, hazard, or vulnerability variable at a time. So we fit 4 exposure-only models. With each index of exposure we calculated from the survey data. Then 6 hazard-only models, 1 with each index of hazard we calculated from the ecological data. And finally, 5 vulnerability-only models. And we used a model fit criterion called the AIC to compare models within each group, and we retained only those variables that had strong statistical support compared to the others. And here I summarize the variables that had that strong statistical support. So for exposure, it was a single index, the simplest of the 4 we looked at, that we retained for both presence and intensity outcomes. A single index of hazard fit best for infection presence, but 4 indices of hazard had comparable fits with infection intensity. Finally, the vulnerability metrics that were retained all related to the use of water and sanitation infrastructure. There were 2 for infection presence and 3 for infection intensity. And with these retained variables, we proceeded to fit models that combined exposure, hazard, and vulnerability. And this resulted in 22 models of infection presence and 40 models of infection intensity. And for each model in each set, we calculated its fit using AIC, and then we used AIC to calculate a model's weight within each set. And this method basically assigns higher weight to models with better fits. We then summed the weights across all models in each set for each component of risk. And these sums will help us understand the relative importance of exposure, hazard, and vulnerability for each outcome. And so summing weights across all models will generate values for each component of risk that range from 0 to 1. And so 0 means that a component of risk was not included in any of the best fitting models, while value of 1 means that a component of risk was included in all of the best fitting models. And so for infection presence, we see that exposure, hazard, and vulnerability are all pretty important. All 3 components appear in most of the best fitting models in this set. But the most important component of infection presence is pretty clearly hazard. It is present in nearly all of the best fitting models. And for infection intensity, we see that all 3 components of risk are all pretty important again, appearing in most of the best fitting models in this set. But the most important component of infection intensity is clearly exposure. And so these findings reflect the different processes that influence whether a person gets infected in the first place, versus how heavy their infection becomes. Exposure was likely less relevant to infection presence because vulnerability assured some baseline level of exposure to a hazard. In contrast, exposure was most important to infection intensity because with more exposure, there's more opportunity to acquire additional worms and develop heavy infections. And so our finding that all three components of risk were important conveys the value of studying schistosomiasis from this sort of integrated perspective of social and ecological processes. And we see that infection, it underscores that infection occurs where the transmission cycle interacts with human activity in time and space. We also found that the relative importance of exposure, hazard, and vulnerability differed depending on whether we were looking at infection presence or intensity as the outcome. This reflects the different processes, as I said, that influence whether a person gets infected and how infected they become. Vulnerability assures some baseline level of exposure, making exposure comparably less important to presence outcomes, while in contrast, exposure is the most important to intensity outcomes because it's how people accumulate worms over time with repeated exposure. We also learned that the best fitting index of exposure was the simplest of the four we looked at. So this involved a single question in the survey as opposed to an entire module of many questions that we're combining and making complex metrics that keep it simple, stupid, right? And finally, we learned that adjusting hazard for distance between a water access site and a household improved the fit of presence models. And so this may actually be an important component of vulnerability, where kids that are living closer to the water are at higher risk of becoming infected. And so from this dissertation, we've learned more about how social factors influence the ecology of schistosomiasis. And in the dammed and endemic landscape of the lower basin of the Senegal River, we learned that people do what they can to avoid contact with water, but they ultimately perceive their risk for schistoinfection to be unavoidable. And in chapter 2, we learned that children whose families cultivate larger areas of irrigated land are more likely to be infected with urogenital schistosomes. These results suggest that the economic opportunities created by the dam add to the risk we know to be elevated by the environmental change caused by the dam. And finally, we determined that exposure, hazard, and vulnerability all contribute to infection outcomes, but the relative contributions vary for infection presence and intensity. These findings highlight the importance of thinking about how both social and environmental processes influence schistoinfection, which is especially true in settings like the Senegal River, where water resources development has changed the environment and how people interact with it. And so adding this social environmental complexity to an already complex life cycle makes things complicated, and it brings us back to this mass drug administration, this simple intervention that doesn't prevent reinfection. And so under this sort of added complexity, how can we expect mass drug administration to address this extra complexity? So my research highlights the importance of implementing complementary interventions that can help interrupt transmission while mass drug administration reduces the disease that already exists in people. And so there's opportunities for creativity and innovation here, in particular involving other sectors to contribute to the fight against schistosomiasis. And so to conclude, I'll highlight some of the ongoing efforts, both globally and among our research team specifically, to diversify the portfolio of strategies used to control schistosomiasis. In 2012, the World Health Assembly passed a resolution pushing member states to implement complementary interventions to mass drug administration. And so such interventions may include the provision of water and sanitation infrastructure, where piped water sources can reduce human contact with the environment, or at least give people the option to not be in the water where the parasites are. And similarly, sanitation infrastructure can contain the eggs that are coming out of infected people and prevent them from making it back into the environment and perpetuating transmission that way. We can also control snail populations and prevent or at least mitigate the risk of reinfection. And while chemicals are often used to kill snails, there are some biological strategies we might be able to exploit in dammed landscapes. So we may be able to restore snail predator populations that have disappeared upstream of dams by retrofitting them with passages that allow these species to move freely across the dam barrier. And the engineering and financing plans are actually in the works for doing this at the Diama Dam on the Senegal River. And this is an intervention that could potentially be implemented across many watersheds in coastal Africa and could reduce schisto risk for a large portion of the population at risk. I have also proposed future work with the Natural Capital Project to develop decision support tools for mitigating schisto risk around dams. So when implemented with MDA, complementary interventions can prevent reinfection while drugs rid people of existing infections. And with this diversified portfolio, I imagine us being able to eliminate schistosomiasis from dammed landscapes by making the environment a safer place for people to live and work. And with that, I think we can transition into Q&A. So if you've submitted a question.
Thank you.
Yeah.
Angela.
Hand it over to Michelle.
Can you hear me?
Yeah.
That was wonderful. It's clear that you showed us that mass drug administration is not enough, that we're going to have a need for an integrated complementary interventions. So thank you very much. I have some questions, but I'm going to start with the people in the audience. Sudhata Ray, you want to read your, introduce yourself and read your question. Thank you. Sudhata?
Can you guys hear me?
Yes.
Andrea, first of all, excellent presentation. I'm one of Andrea's colleagues at E-IPER in the same year. My question was, so in your second chapter where you didn't find intensity, like any sort of relationship with children from agricultural households and intensity, I was wondering, did you look at the time spent by the children in agricultural fields? And if that had any relation to intensity of infection?
Yeah, so we collected some data on children's participation in agriculture, but this ended up being a really sparse piece of data statistically. Very few families reported kids' participation in a variety of agricultural activities. And so when we're looking at area of irrigated land, this is sort of an indirect measure. So we're not looking at behavior specifically, but just the how much land families are owning. Yeah, and so while we tried to, we wanted to, we thought about collecting data on kids' participation in agriculture. We weren't able to use those data in the analysis just because so few children reported participating, or so few families reported children participating in agriculture.
Okay, Samantha asks, did you feel like there was enough heterogeneity in vulnerability across the community? And what types of people in the community are less vulnerable to schisto?
Yeah, so that's a good question. Thanks, Sammy. We actually, the two vulnerability metrics that we ended up looking at in the combined analysis was had to do with water and sanitation infrastructure, and actually the dependence of households on surface water for a couple of different activities was split pretty evenly between yes and no for households. And so there was about half the households didn't report any using surface water for any of the two chores we considered, while half reported either one or two activities. And so that, I think we had tighter confidence intervals on those estimates than we did for sanitation, which was much harder statistically, or wasn't as evenly distributed. Most households had some sort of sanitation. There was very few households without sanitation. And so what types of people in the community are less vulnerable to schisto, I would say would be people who are not using water and have the means to reduce their exposure.
Kate, do you want to ask your question?
Hi, my name is Kate Lindsay. I'm one of Andrea's friends and colleagues. And my question was, I was a little surprised at the difference between the urogenital and intestinal infection presence with the irrigation habitat. And I wonder if you had any ideas why you would see one have a strong correlation and the other one not. And then my other question is just a fun one, is if there's anything that came out of the household surveys that really surprised you, like something that you learned during those surveys. Thanks.
Thank you, Kate. To answer the first question about the differences between urogenital and intestinal presence with the irrigated area, the two parasites are transmitted by different genera of snails, and so that have actually quite different ecologies. And so I think this would be an interesting area for future research to pursue in the future, is thinking more explicitly about the snail ecologies and which snails we find in irrigation canals, for example. But I also think that there's potentially some the way that the infections are analyzed. So we have urine samples for urogenital schistosomiasis, fecal samples for intestinal schistosomiasis, and we analyze urine samples using urine filtration, and then fecal samples using this it's called CatoCats smear, and the diagnostic kind of like ability to detect low intensity infections using that CatoCats smear for intestinal infection is actually quite low. And so I think that the weakness of that diagnostic may have influenced our ability to detect the low intensity infections. So I think that could play a role, but I also think that the snails may play a role. And then the other question was about what did I learn from the survey data? And I think one of the things that I took away, we collected some data on fertility and childbearing among women. And so as I was going through surveys, doing data checks and stuff, I almost always took note of like how long women in Senegal are bearing children for most of their lives. And that just always blew me away. And it being very different from my reality, where I'm a 32-year-old woman who doesn't have kids. That's just to put that into stark relief.
So, Garrett, well, we have an anonymous attendee, so I will ask this. This appears to be an area that may have other waterborne pathogens. Does comorbidity play a role in the progress of schisto? Is this important for future studies?
I'm not sure. I don't think I can address the question about progression of disease, but there are absolutely comorbidities that happen, and there are lots of things that interact with each other. And so, for example, schisto, urogenital schisto is known to cause something called female genital schistosomiasis, where lesions are developing in the genital tract of women. And there's evidence coming out that this is likely increasing risk for HIV. And so there's lots of really important and interesting interactions going on between different parasites and pathogens in this setting. There are likely diarrheal pathogens that are probably playing a role, affecting like nutritional outcomes and things like that, and polyparasitism that is, kind of building on each other. But teasing all of those apart has been really challenging. People are doing it, but it's an interesting and challenging area to work.
So we only have 2 more minutes, and Gary, you have a multi level question. You want to shorten it a little bit and ask Andrea her last question, and then we'll go into our closed door session.
My question, dear Andrea, is I'm wondering about the effects of the changes in population density and how this is going to affect prevalence going forward. And are there societal or systemic ways of dealing with that, given that we're likely going to be seeing increased population and population density over time there?
Yeah, that's a big question, Gary. Thank you for asking it. Population density is thought to have played a role in schistosomiasis in the Senegal River. It's a generally rural place. There's not a high population density, but the construction of the dams was attractive because there was all of this agriculture that was getting developed, and there were opportunities for people to work. And so there are hypotheses out there that population density was contributing to the outbreak of schistosomiasis we saw in the lower basin of the Senegal River, and the Senegal River basin in general. But I think that there are some studies that have borne that out to say that, you know, the increases that we saw were in spite of this population density that was also encroaching. And this goes a little bit beyond the system of schistosomiasis specifically, but there are absolutely, you know, kind of human interactions with the environment, human interactions with wildlife that are playing a role in you know, the spillover of pathogens with, you know, our current pandemic with coronavirus. And so human encroachment on like wildlife habitat is likely an important thing for us to be considering and mitigating the risk for those types of spillover events. So I want to start by just acknowledging the funding, for which I'm very grateful for. So I had funding both from E-IPER and the Stanford Interdisciplinary Graduate Fellowship Program. These donors, the donors of these scholarships gave me tremendous freedom to pursue research I cared about on my terms, so thank you. I'm also grateful to the Bill and Melinda Gates Foundation and the National Science Foundation, which supported the larger project in which my research was embedded. This funding allowed me to oversee an enormous data collection effort, which was hugely valuable experience for me professionally. And this funding also gave me access to the parasitological and ecological data that I used in chapters 2 and 3. I worked with 2 tremendous teams of enumerators and supervisors in Senegal to collect the survey and focus group data that I reported on today. We spent long hours together in these study villages, driving on bumpy roads up and down the lake, eating rice and fish from communal bowls every day for lunch, and spending lots of time away from our families. I've learned so much from these teams. I learned to take a tea break after lunch and chill out. I learned about the importance of quick feedback loops on data quality. I learned how to respond to unsolicited marriage proposals from villagers. And most of all, I learned so much about our study population from the data these folks painstakingly collected. And the leadership at EPLS also played an enormous role in making our research happen, including Gilles Revaud, their CEO, and Nicolas Journad, the project manager. These 2 and everyone else at EPLS welcomed me warmly in Senegal. Nicolas even went out of his way to show me where the grocery stores were, and allowed me to spend a summer living in a spare room in his house. They and the administrative, logistics, and data management staff took care of so many details, hiring the enumerators, maintaining vehicles, printing out hundreds upon hundreds of multi-page surveys, and their work on these details helped me to focus on the data and its collection and its quality. Whoops. Okay. I also have many people to thank at Stanford. First, my committee. Thank you for giving me both the space and the guidance to pursue my research. I've benefited tremendously from taking ownership of the design, data collection, analysis, and interpretation of each chapter in my dissertation, but my work has benefited tremendously from your engagement with it too. Giulio, my research has often fallen outside of your precise methodological expertise, but you've never wavered in your support for me or the researcher I wanted to become. And when you came to Senegal to visit us in the field in 2018, I gave you a hard copy of the draft of chapter 1 I had been working on and asked you to read it. The clear-eyed feedback you gave me that day, in which you saw not one terrible narrative, but 2 interesting ones, gave me confidence to forge onward and tell those stories. And Jason, my single-minded focus on schistosomiasis in Senegal came at the cost of us working more closely together on a project. But you've always made yourself available to me, and I've benefited greatly from your pragmatic perspectives on research and career. The thoughtfulness with which you pursue research in your lab will always be an example I strive to emulate.
Sweet.
David Lopez-Carr, when I visited you as a prospective student in geography at UCSB, it was pretty apparent that we would be working together regardless of which school I chose to attend. And even then, you were prepared to entrust me with the responsibility of managing the on-the-ground activity of the NSF grant. That felt like a lot to take on, and it was, but I have learned so much from the experience. Thanks for your confidence in my abilities and the pep talks you have given me along the way. And David Rehkopf, I'm so glad I took your course in social epidemiology. At the time, it helped me to make connections across my fields of inquiry that I was developing for my quals proposal. And since then, I've been really grateful for your willingness to dig into the details of the epidemiologic methods of my research, especially in chapter 2. You've made my work stronger. Thank you. And finally, Michelle, thank you for chairing today. The defense would not have been possible without you, but I'm honored to have your vast experience in global health present today too.
It's a pleasure. All right, everybody can use the clap thing on the, let's see, can we get a gallery clap?